Our today's lecture topic is plant tissue culture. First, we will see what is plant tissue culture. Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile condition on nutrient medium or MS medium. And it is widely used to produce clones of a plant in a method known as micropropagation. Let us see the brief history of plant tissue culture. So, the idea of totipotency was given by Haberland in 1902, and in 1937, this root culture of tomato was first established successfully by White. Then, in 1941, this Vancouver Beek used coconut milk to grow and develop the young Datura embryos. Then in 1957, Skoog and Miller demonstrated the role of auxin and cytokinin on root and shoot formation in tobacco tissue. In 1962, Murashige and Skoog was introduced the medium for tobacco culture. Let us see the different techniques in plant tissue culture which shows their advantages over the traditional methods of propagation, and this method, plant tissue culture methods, how they are more advantageous over the traditional method. So let's begin. The production of exact copies of plants that produce particularly good flowers, fruits, or they have other desirable traits. And to quickly produce mature plants means by using this plant tissue culture, we can shorten the life cycle of plants. Then the production of multiples of plants in the absence of seeds and necessary pollinators to produce the seeds means the plant varieties which are sexually incompatible, they can also be grown or reproduced by using this tissue culture technique. Then the regeneration of whole plant from plant cells that have been genetically modified. So the plants which are made by the recombinant DNA technology can be regenerated into entire plant by using the plant tissue culture technique. Then the production of plant in sterile containers that allows them to be moved with greatly reduced chances of transmitting the disease, pest and pathogens means uh, sterile plantlets which are free from any kind of diseases or pathogens or pest can be regenerated uh, in vitro condition by using the plant tissue culture technique. And the production of plants from seeds that otherwise have very low chances of germinating and growing, that is like orchids and nepenthes. These plants means orchid and nepenthes, they have low chances, their seeds have low chances of germination and growing in normal conditions. But if we use plant tissue culture technique, we can grow them easily, or we can produce plantlets from these kind of seeds very easily. Then let's see the next point, that is to clean particular plants of viral and other infections, and to quickly multiply these plants as clean stocks for horticulture and agricultural practices, we can use plant tissue culture techniques means here the virus free plantlets or plant which are produced, they are free from any kind of infection, and these type of sterile plant or clean stock, we can call them as clean stocks, they can use for horticulture and agriculture purposes. Let us see the flow chart of procedure of plant tissue culture in brief. So for beginning the plant tissue culture procedure, we require one healthy mother plant. So intact plants are required first, and small excision of plant or small portion of plant which we use to inoculate, it is called the explant. So this explant, uh, first we should choose the healthy mother plant, and then take excision of that explant, and after that the proper trimming of explant should be done, and after that surface sterilization is the most important technique. In this technique we require various surface sterilants like HGS, so HgCl2, 70% ethanol, and many other sterilants you can use. Then sterilization of glassware, it is also one necessary step involved in plant tissue culture procedure. Then explant is washed in sterile distilled water. After that, the culture medium is sterilized in autoclave. Before that, we should prepare the MS medium first. MS medium. What is MS medium? It is the Murashige and Skog medium, and it provides the carbon nitrogen sources and some vitamins, some amino acids, and some phytohormones. These are included in that media, and they support the plant growth in vitro condition. Then after that, the inoculation of explant in the culture medium under aseptic condition. So in laminar airflow, we can transfer the explant on the MS media containing bottle. We can use culture bottles or we can use culture tubes for this purpose. Then after that, uh, incubation under suitable conditions for proper culture is required. Incubation means allow them to grow in suitable conditions. Here, which kind of suitable conditions we require? We require 25 degrees Celsius temperature, then 2000 lux light intensity, 16 by 8 photoperiod, then 60% relative humidity. These are the parameters which we require to grow plants uh, in vitro. So after that, subculturing. After getting the callus, after a few weeks we will get callus, and after getting callus, subculturing is required. So after subculturing means what? Subculturing means the transfer of that callus on freshly prepared media, which contain appropriate concentration of auxin and cytokinin. For example, for shoot growth, we require cytokinin in higher concentration and auxin in lower concentration. And for root regeneration, we require auxin in maximum concentration and cytokinin in lower concentration. And after transferring this callus or this plantlet on suitable MS media, after a few weeks we will get entire plantlet, and these plantlets are ready for hardening purpose. Hardening means acclimatization. So let us see what is acclimatization. Acclimatization is the process in which we grow the plantlets in soil, but under certain controlled condition before transferring them into harsh condition which are present in the field. So these are some steps, general steps or techniques involved in plant tissue culture. Then here we require the MS media. As I have told you, MS media is required in plant tissue culture. So this MS media, it contains some micronutrients, macronutrients, then some vitamins, amino acids, inorganic salts, and phytohormones also. So micronutrients, uh, here we require or we add the elements like iron, then manganese, zinc, etc., and also crucial for the growth of tissues. Then carbon source is required, means carbohydrates are also required, and here for carbon or energy source we require the sucrose. Then macronutrients, here nitrogen or phosphorus, potassium, calcium, sulfur, these type of micronutrients are required by the plant for their growth. uh, then some vitamins are required, some amino acid like glycine is required here, and some phytohormones are required like auxin, indole acetic acid, then indole butyric acid, then cytokinins are required, and gibberellic acid can also be used in combination. Then let us see the steps involved in plant tissue culture. We have already seen that, but here I want to summarize it. So selection of an explant, then surface sterilization, then preparation of MS media, then inoculation of explant on MS media, incubation, then callus formation, then subculturing, then incubation, then formation of plantlets, and then acclimatization, and then transfer in the field. Then here pictures showing the callus formation culture room where we want to incubate the culture bottles, and then uh, here we can see the type of culture bottles and culture tubes which we required for growing the plants. Then let us see the types of plant tissue culture. There are various types of plant tissue culture techniques. It includes the seed culture, embryo culture, callus culture, organ culture, protoplast culture, and anther culture. Let us see the advantages of plant tissue culture. So first advantage is formation of large number of clones from single explant. Means we can prepare thousands of plants by using single explant, or a small portion of plant we can use for growing millions of plants. Then second advantage is the selection and culturing of desirable traits. We can produce desirable varieties by using plant tissue cultures. Means if you want to produce plant with higher chlorophyll content or higher protein content or any other trait, disease resistance variety you want to develop, you can develop such kind of traits by using plant tissue culture. Then production without seed, means for regeneration of plant, here we don't require the seed. So plants which cannot bear seeds, they can also be regenerated by using this plant tissue culture technique. Then this plant tissue culture is season independent technique. Means by providing particular environmental conditions, in vitro condition, we can produce the plant in any season. So last advantage, short production time and long generation time. Means here we can shorten the life cycle uh, of plant. Means in shorter duration we can take the yield. So this is all about the plant basics, or it is beginning of plant tissue culture. In next video lecture we will see how it works. Thank you.